Good day, future doctors. So this is the fourth lecture in our series of lectures for trematodes. For today, we will be talking about the hepatic flukes. Before I will mention the names of the hepatic flukes, it's very important to be reminded that even if they are named hepatic flukes, the adult worms of these parasites are not found in the liver parenchyma. They are particularly found within the bile ducts. The presence of the worm within the liver parenchyma would only indicate possibility of severe infection. These are our hepatic flukes. We have Fasciola hepatica, Fasciola gigantica, Clonorchis sinensis, Ophistorchis philenius, and Ophistorchis vivereni. We will now discuss the life cycle of our first two hepatic flukes, the Fasciola hepatica and the Fasciola gigantica. The adult worms, as indicated in the picture, are found in the biliary ducts. Once the female worms will lay eggs, the eggs will be released into the bile and will be drained into the duodenum, so expect the eggs will be discharged through the feces. I hope you can remember that all trematodes would lay embryonated ovum except for PEFF, and one of the letter F's in there is referring to fasciola. That's why according to the number one part of the life cycle, what is discharged out from the feces of the affected human host is the unembryonated ovum, and this is also the diagnostic stage. Before I will proceed with the life cycle, it's important to take note that since humans harbor the adult stages of the parasite, so humans act as definitive hosts. Now, this unembryonated ovum should stay for days in the water before it will embryonate and release now the ciliated stage, which is the meracidium. The meracidium will infect the first intermediate host, which is a snail. Once inside the body of the snail, the meracidium will transform to form the sporocyst. The sporocyst will form the feeding stage, the rhidia, and the rhidia will eventually release the tailed stage, cercaria, and the tailed stage, cercaria, will come out of the body of the snail. And this tailed stage cercaria will infect the second intermediate host. If in the case of Paragonimus, the second intermediate host is a crab or a crayfish, in the case of Fasciola, it's an aquatic plant. So, the cercaria will insist itself to form the metacercaria in aquatic plants. Eating raw aquatic plants will now predispose the human host to develop the infection because the human host will get the infective stage, which is the metacercaria. Now, we have two species of Fasciola. Fasciola hepatica basically infects sheep. Fasciola hepatica, Fasciola sheepatica. And Fasciola gigantica usually infects cows. Gigantica, and cows are bigger than sheep. This is a picture of Fasciola gigantica and Fasciola hepatica, and take a look at the difference in the size. Fasciola gigantica commonly infects cows, while Fasciola hepatica commonly infects sheep. Again, even if they are referred to as hepatic flukes or liver flukes, the adult worms of Fasciola, Clonorchis, and Ophistorchis are found within the bile ducts. Now, if the infection will become chronic or severe, there's a probability that the worms will start to appear also within the liver parenchyma. Now, these are the common names of the two species of Fasciola. Let's start with Fasciola hepatica. Since it commonly infects sheep, it is also known as the sheep liver fluke. And it's also popularly known as the temperate liver fluke. So for you to easily remember, you have two letter E's in the word sheep, right? So I intentionally typed in the letter is as capital letters so that you can easily memorize that the other name for Fasciola hepatica is sheep liver fluke, Fasciola shepatica. And since there are three letter is in the word temperate, you can also memorize it easily. Fasciola gigantica, since it's bigger than Fasciola hepatica, it's also known as the giant liver fluke. It is also known as the tropical liver fluke. If you are shown a picture of a trematode and you want to know whether it is Fasciola or not, you just simply have to look for one distinguishing feature, and that is the presence of the cephalic cone. That's the extension that you can find on the anterior end of the worm. I want you to take a look at the pictures of, from the left, Fasciola, middle, Paragonimus, right side, Schistosoma. Fasciola can easily be identified because of the presence of its cephalic cone, which you do not see in Paragonimus worm. Please remember the Paragonimus worm has a coffee bean shape and its ventral sucker is found in its midline, while the ventral sucker of Fasciola is found near its oral sucker. Schistosoma can easily be differentiated from other trematodes because it's not dorso-ventrally flattened and it is dioecious. This is the picture of the Fasciola worm. Take a look at its cephalic cone, and then this picture will also remind you that among the trematodes that we will cover, it is only the Fasciola that has a branch intestine. This is another picture of Fasciola worms. Please do appreciate the presence of cephalic cone and the location of the ventral sucker. It's located near the oral sucker. While in Paragonimus, the ventral sucker is located in the midline. Aside from the size, there's also one unique feature that can help us differentiate Fasciola hepatica from Fasciola gigantica, and that is the presence of the shoulder, that structure pointed by the blue arrow. The shoulder is prominent in Fasciola hepatica, while it is less evident or less prominent in Fasciola gigantica. Both of these worms have cephalic cones, so they belong to the Fasciola species. Which among them do you think is Fasciola hepatica? Which among them is Fasciola gigantica? Letter A is Fasciola hepatica because it's shorter than Fasciola gigantica, the worm in letter B, and it has this prominent shoulder. Now, let's try to label the parts of Fasciola species. This is the oral sucker, and this is the pharynx, and this is its intestine. Please do appreciate that the intestine is highly branched, which is very different from the simple and unbranched intestines of the other trematodes. And this is the ventral sucker, which is located near the oral sucker. Let's try to identify the other parts of the worm. This is its uterus. This is its single branched ovary. These are two of its testes, and they are located on the posterior part of the worm, while the ovary is found anterior to the paired testes. And these testes are branched or dendritic. And then, on the lateral part of the worm, you have there the vitellaria, and it extends from the shoulder down to the posterior end. At this point, I want you to identify the structures pointed by the following arrows. Can you please identify the structure pointed by the red arrow? Yes, that's the oral sucker. How about the structure pointed by the orange or brown arrow? That is the, yes, oral sucker. And then, the structure pointed by the green arrow. That is the vitellaria. And it extends from the shoulder down to the posterior end. How about the blue arrow? It's pointing to one of the testes of the worm. And it is described to be branch or dendritic. How about these next sets of arrows? So red arrow is pointing to the structure that is connected to the oral sucker. What do you think is that one? That is the pharynx. Very good. White arrow is pointing to the branch intestine. Very good. How about the green arrow? That's pointing to the uterus. And the blue arrow is pointing to the single branch ovary of the worm. So let's try to review the descriptions of the following parts of the fasciola worm. Intestine is branch, while most of the trematodes have simple or unbranched intestine. Testes are paired and they are branched or dendritic. The single ovary is branch, while the vitellaria is from the shoulder down to the posterior end of the worm. So this will summarize to us the descriptions of the different parts of the fasciola worm. And it's very important to highlight that this is the only parasitic trematode that has cephalic cone and the shoulder. So what are now the distinguishing characteristics or features of fasciola worms? We have the presence of the cephalic cone, the shoulder that is found on both sides of the cephalic cone, and the branch intestine. Please recall that all parasitic trematodes lay embryonated ova except for PEFF, Paragonimus, Echinostoma, Fasciola, and Fasciolopsis. And I hope you can still remember at this point that the eggs of these 4 trematodes are difficult to distinguish from each other except for Paragonimus. I hope you can still remember what characteristic of the Paragonimus ovum that will help you identify it from the ova of Echinostoma, Fasciola, and Fasciolopsis. The description of Fasciola hepatica ovum is almost similar to that of Paragonimus. It is ovoid in shape, it is yellowish to brown in color, it is operculated, and it is unembryonated upon discharge. What sets it different from Paragonimus ovum is that its posterior end is not thickened. Now, since the adult worms are found within the bile duct, the female worm will lay the eggs in the bile duct and the bile will drain into the duodenum. So expect that the diagnostic specimen for the diagnosis of fascioliasis is the feces or stool. Again, yellowish to brown in color, ovoid in shape, operculated, but the posterior end is not thickened unlike with Paragonimus ovum. The problem now is it would be hard for us to distinguish this ovum from Echinostoma ovum and Fasciolopsis ovum. That's why it's important to get the medical history of the patient. We need to ask the patient, are you eating raw aquatic plants? And that will somehow support our impression that the patient is having fascioliasis or infection with fasciola. This is just to review you guys on the microscopic characteristics of the Paragonimus ovum. It is ovoid in shape, similar to that of Fasciola ovum. It is yellowish to brown. It is operculated. It is unembryonated, but it has a thickened posterior end. And it's very important to differentiate Paragonimus from Echinostoma, Fasciola, and Fasciolopsis ovum because Paragonimus ovum, even if the adult is found in the lungs, can still be found in the stool sample. What are now the first and second intermediate hosts of the Fasciola species? For the snail hosts, they infect the Limnaea species of snails. And these snails are most of the time found in water. That's why it's easy for the cercaria that will emerge from the snail to infect the second intermediate host, because the second intermediate hosts are aquatic plants. So, the first and the second intermediate hosts are found within the same environment. So, what are the examples of this second intermediate host or aquatic plants? We have Ipomea obscura and Nasturtium officinale. Ipomea obscura is also known as kangkong, while Nasturtium officinale is popularly known as the watercress. These are the Limnaea snail host of Fasciola. They stay most of the time in water. And these pictures will just show us that the snails are living on the same environment as the second intermediate host. So, the cercaria will just easily leave the bodies of the snails and transfer to the aquatic plants or the second intermediate host. The second intermediate host would include the Nasturtium officinale or the watercress and Ipomea obscura or popularly known as kangkong. Eating raw aquatic plants will put you at risk of developing fascioliasis or infection with Fasciola gigantica or Fasciola hepatica. But there's a plot twist. Do you know that the cercaria of Fasciola can exist while floating in the water? So, in some instances, they don't need the presence of the second intermediate host. If that's the case, the mode of transmission is drinking water with the metacercaria of the Fasciola species. So therefore, there are two ways on how we can get the fascioliasis or Fasciola infection. Eating raw second intermediate host or the aquatic plants, or drinking water with metacercaria stage. Human infection begins with the ingestion of the metacercaria stage. Once present in the intestine, do you know that this metacercaria or immature fluke will penetrate the wall of the intestine because they are not supposed to live there? We're talking about hepatic flukes and they're supposed to live as adults in the bile duct. So, the immature fluke or the metacercaria will penetrate the wall of the intestine to escape into the peritoneal cavity and migrate towards the capsule of the liver. We call it as the Gleason's capsule. So they will dig their way into the Gleason's capsule to gain entrance into the hepatic parenchyma or tissue, and in order for them to gain access into the bile ducts. The early stage of the infection is referred to as acute or invasive phase because there is the invasion of tissues by the immature fluke or the metacercaria. The penetration of the metacercaria through the intestinal wall will not cause any significant problem. Their presence in the peritoneal cavity may still be asymptomatic, but the moment that they will burrow themselves into the Gleason's capsule, into the hepatic tissue, for them to gain access into the bile duct, that's the time that the patient will become symptomatic or will manifest the signs and symptoms. And what are these? We will discuss that later. The acute or invasive phase of fascioliasis can also be referred to as the migratory phase because it's primarily because of the migration of the metacercaria or the immature fluke. It can also be called as invasive phase because there's invasion of the Gleason's capsule, of the liver parenchyma, and of the bile duct. Hepatic phase, I know you can already explain in your minds why. It can also be called as the parenchymal phase because there's invasion of the parenchyma of the liver. And it's also called as the larval phase because what is migrating is the immature metacercaria or the immature larva of the Fasciola species. Now take note, it is in the early stage or the acute or invasive phase of fascioliasis that the patient will exhibit signs and symptoms. And what are these? high fever because there is infection. Hepatomegaly because there is damage to the liver brought about by the invasion of the immature metacercaria or fluke. And since you have here the presence of a parasite, expect that the patient will have eosinophilia. That's why if you will encounter a patient in the future complaining of right upper quadrant, you might also want to test the CBC of the patient and check for eosinophilia. But before you do that, you really have to get a good history of the patient. You have to ask the patient, are you eating raw aquatic plants? Or have you been to places where the fascioliasis cases are very high? In acute or invasive phase, there is larval migration through the liver parenchyma, resulting to tissue destruction, resulting to inflammation, resulting to internal bleeding. That's why some patients would even develop anemia. And do you know that some patients, aside from exhibiting the fever, the eosinophilia, the hepatomegaly, some of them would complain of urticaria because they are allergic to the chemicals produced by the worm. And this acute or invasive phase can last for 3 to 4 months. During the acute or invasive phase, the metacercaria or the immature fluke is penetrating through the Gleason's capsule of the liver into the liver parenchyma and so that they can reach the bile duct. Do you know, once these worms will reach the bile duct, the patient will progress from acute or invasive phase to chronic phase. And the patient, most of the time, will become asymptomatic. So if the patient would have fever, right upper quadrant pain due to fascioliasis, and if the patient will not seek treatment, the patient will just assume that he had already recovered from the disease because he will become asymptomatic in chronic or latent phase. This phase will only become problematic or the patient will just become symptomatic in the presence of severe infection because that's the time that the worm could cause obstruction, bile duct obstruction, fibrosis, and in some cases, the walls of the bile duct might erode, allowing now the worm to be reintroduced into the liver parenchyma. And the worm there will cause destruction and later on cause a large abscess formation. In the chronic phase, the immature larva will be able to reach the bile duct and mature there as adults. The adults will mate and the female will lay eggs and the eggs will be released into the bile, then into the duodenum only to be excreted in the feces. Most of the time, especially in light infections, patients do not exhibit any sign or symptom. And the only lab result that will be positive during the chronic or latent phase is the CBC. And the CBC will reveal eosinophilia. CBC or blood smear examination. And this is the reason why patients will only exhibit the signs and symptoms in the acute or invasive phase. Because during that phase, fasciola and its antigens can induce cytokine production by T helper 1. That's why there is immune reaction against the parasite. The patient will become symptomatic. But later on, especially in the chronic phase, the parasite can stimulate the T regulatory cells to produce interleukin 10. And interleukin 10 is an immunosuppressive cytokine. So therefore, it can suppress the immune responses. Thereby, the patient will not exhibit any sign or symptom. This is the liver, and take a look at the worm pointed by the blue arrow. It has a cephalic cone, and that will only confirm to us that fasciola is indeed a hepatic fluke. This is a picture of a liver with 5 fasciola worms. In some Mediterranean countries where livers of sheep and goats are eaten raw, ingestion of the raw liver may cause suffocation. Why? You know very well, you have there in the liver of the sheep or goat the adult worms. And these adult worms may be temporarily lodged in the pharynx, causing now obstruction. Eating raw livers of goats and sheep can cause what we call pharyngeal fascioliasis. Adult worms may be temporarily lodged in the pharynx, causing now obstruction. And pharyngeal fascioliasis is also known as Halzone. Halzone or pharyngeal fascioliasis happens after the ingestion of the raw livers of the infected sheep or goat. The adult worm present in the liver of the infected sheep and goat might be lodged into the pharynx, causing now infection, swelling, and obstruction. That's why patients with this condition would complain of dyspnea, dysphagia, and even deafness. Why? Because sometimes the worm can gain entrance into the eustachian tube, which connects the nasopharynx to the middle ear. There is also what we call false fascioliasis, and this also occurs after the ingestion of the raw liver of infected sheep and goat. If we eat the liver raw, there's a chance that we will be ingesting the eggs of the adult parasites present in the raw livers. And what will happen? Their eggs will be passed out through our stool. And the presence of the eggs in the stool might misdiagnose us as a case of fascioliasis, when in fact we don't have the infection. It's just that we were able to ingest the eggs from the raw liver that we have eaten. Please do recall that the metacercaria in the duodenum will pierce through the wall of the intestine into the peritoneal cavity. And there's a high chance that the worm might get lost and will not invade the liver. Instead, they will infect other organs. And we call that condition as ectopic fascioliasis. Ectopic infections with fasciola are infrequent but can occur in the peritoneal cavity, intestinal wall, lungs, especially if they will pierce through the diaphragm to gain access into the pleural cavity. Some of them can even reach the eye and the brain. This is a case of ocular fascioliasis. You can clearly see within the eye of this patient the adult worm with the cephalic cone. This is a case of cutaneous fascioliasis wherein the affected organ is the skin. Do you know that if we will try to biopsy this skin lesion, we will be able to harvest one flat worm that is unsegmented and it has a cephalic cone. Fasciola gigantica is bigger than that of Fasciola hepatica. And the infection with Fasciola hepatica is common among sheep-raising countries. In the case of Fasciola gigantica, its cases are high in countries where people would raise cattle and water buffaloes. Both worms have cephalic cones. To differentiate Fasciola hepatica from Fasciola gigantica, you look for the prominence of the shoulder. Do you know that the only structure that is prominent or bigger in Fasciola hepatica compared to Fasciola gigantica is just its shoulder? Because the other structures or parts of the worm are bigger or more prominent in Fasciola gigantica. Fasciola gigantica is larger. It has a larger ventral sucker. Its intestine, ovary, and testes are more branched than that of Fasciola hepatica, and its ova are generally bigger. While the only structure that is More prominent in fasciola hepatica is the shoulder. The treatment of choice for fascioliasis is triclabendazole. An alternative drug is bithionol. And what do you notice? So far, for the first two trematodes we have discussed, praziquantel is the drug of choice, but it's not recommended for fascioliasis. Now, how to prevent fascioliasis? Washing or cooking of vegetables, because the second intermediate host is an aquatic plant. Boiling of drinking water, because there's a chance that the metacercaria will just be freely floating in the water and it's not encysted in the second intermediate host. We can also eliminate the snail host to control the number of cases, and it's very important to treat positive cases so that they will not add on to the number of new cases. Now, we're so done with the first two hepatic flukes. It's now time to go to the last three. We have the Clonorchis sinensis, Ophistorchis philenius, and Ophistorchis vivereni. This is the life cycle of Clonorchis sinensis, and I want you to listen very carefully. The adult worm is found in the biliary tract or bile duct. And the adult females will lay eggs into the bile duct, only to be drained into the duodenum, and the egg will be discharged through the feces of the affected human host. Just like in the case of the other trematodes, humans act as definitive hosts in the life cycle of Clonorchis sinensis. Now, it's very important to take note that the ovum of Clonorchis sinensis is discharged as embryonated, and their snail hosts are terrestrial type. That's why these embryonated ova should be washed first into the shore so that they can be accidentally eaten by the first intermediate host, which is the snail. That's why it's very important to remember that it is not the meracidium stage that is considered as infective to the snail host in Clonorchis and even to Ophistorchis. The infective stage to the snail host is the embryonated ovum. Once ingested, the egg will open up its operculum, releasing now the meracidium, and the infection of the snail will now start or take place. So the meracidium will become the sporocyst. The sporocyst will give rise to the rhidia, and the rhidia will produce the tail stage cercaria. And that's the stage that will emerge out from the body of the snail host. Please describe the cercaria of Clonorchis. I forgot to describe the cercaria of fasciola, but I want you to pause this video and check the life cycle of fasciola and appreciate that its cercaria is Lophocercus because it has a long tail. In the case of Clonorchis, it's also a Lophocercus type because the cercaria has a long tail. Now, in the case of Paragonimus, the second intermediate host could be a crab or a crayfish. In the case of fasciola, it's aquatic plant. In the case of Clonorchis and as well as the Ophistorchis, the second intermediate hosts are freshwater fishes. And in the body of these fishes, the cercaria will transform to form the metacercaria. And human infection begins with the ingestion of raw fresh water fish with the metacercaria stage. This is the life cycle of Ophistorchis, and it's basically similar to that of Clonorchis sinensis. The adult worm is found in the bile duct. So therefore, in this case, humans act as definitive hosts. What is discharged from the body is the embryonated ovum, and this is supposed to be ingested by the terrestrial snail. So therefore, the infective stage to the snail is the embryonated ovum and not the meracidium. The meracidium will be released inside the gut of the snail host, and the miracidium will become the sporocyst. The sporocyst will give rise to the rhidia, and the rhidia will produce the tailed stage, which is the cercaria. That's the one pointed by the red arrow. Please classify the cercaria of Opistorchis. Yes, it has a long tail, and the long tail is provided with a fin. So therefore, this is not just a Lophocercus type. This is a Pluro-Lophocercus cercaria. So let's review. Paragonimus has Microcercus cercaria. Schistosoma has Furcocercus cercaria. Fasciola has Lophocercus cercaria. Clonorchis also has a Lophocercus cercaria. And the only trematode that we will discuss that has a Pluro-Lophocercus cercaria is Ophistorchis. And just like in the case of Clonorchis sinensis, the second intermediate host is a freshwater fish. And the cercaria will insist in the body of the fish as metacercaria. Let's review the characteristics of some parts of the fasciola worm. The intestine is branched, the only parasitic trematode to humans that has a branched intestine. The paired testes are branched or dendritic. The single ovary, which is found anterior to the paired testes, is branched or dendritic. The vitellaria is found on the lateral part of the worm, and it starts from the shoulder down to the posterior end of the worm. And you have there the cephalic cone with the shoulder. This is the adult Clonorchis sinensis, and I want you to check its vitellaria. Can you appreciate that it is only found within the middle 1/3 of the worm? That's one of the things that you can rely on in identifying the Clonorchis adult worm. Now, let's discuss the individual parts of the worm. Red arrow is pointing to its oral sucker. Green arrow is pointing to the pharynx. This inverted Y-shaped structure is its intestine, and it's very obvious it is simple or unbranched. Let's check the other parts of the worm. This is its vitellaria, and it's only located within the middle 1/3 of the body. So this can help you in identifying Clonorchis adult worm. Though, this characteristic is also true to Ophistorchis adult worm. So later on, I will teach you how to identify Clonorchis and Ophistorchis adult worms given that they both have vitellaria only in their middle 1/3 of the body. Next, this is its single ovary, and it's very obvious it's lobulated. Then this is the brown colored uterus. And then I want you to take a look at the paired testes. They are branched or dendritic. So let's fill in the blanks. The intestine of Clonorchis is simple or unbranched. There's only one trematode worm with branched intestine, that's Fasciola. The testes are branched or dendritic. The single ovary is lobulated, while the vitellaria is only found in its middle 1/3. So these are the answers to the fill in the blanks question. Please take note, Clonorchis sinensis is also known as the Chinese liver fluke. Clonorchis sinensis, okay? And then the worm is 10 to 25 millimeters long by 3 to 5 millimeters wide. And let's try to compare the size of Clonorchis to that of Ophistorchis later on. Just remember these numbers, 10 to 25, 3 to 5. This is the Ophistorchis adult. What do you notice? Yes, the vitellaria is also found within its middle 1/3, similar to Clonorchis adult worm. Let's identify the parts. This is the oral sucker. This is the pharynx. This is the intestine, and it is also simple or unbranched. Let's identify the other parts. We have the vitellaria, which is similar to Clonorchis. It is only found within the middle one-third of the body of the worm. This is its single ovary, and it is lobulated or lobed. This is its uterus. So up to this point, no, basically all the structures that we have mentioned are similar to both Clonorchis and Opistorchis. Do you know what will help us identify whether the worm is Clonorchis or Opistorchis? Are the paired testes. The paired testes of Opistorchis are lobulated, while the paired testes of Clonorchis sinensis are branch or dendritic. So let's fill in the blanks. The intestine of Opistorchis is unbranched. The testes are lobulated in contrast to the branch testes of Clonorchis. The single ovary is lobed or lobulated, while the vitellaria is only found within the middle one-third of the body. So these are the answers to the fill in the blanks. And Opistorchis adult worms are generally smaller than that of Clonorchis because the Clonorchis worm can be as long as 10 to 25 millimeters, while this worm can only be as long as 8 to 12. Clonorchis sinensis can be as wide as 3 to 5. This one can only be as wide as 1.5 to 3 millimeters. I put here side by side the Clonorchis and Opistorchis adult worms. Both have vitellaria situated in the middle one-third of their body. Please take note that the vitellaria of Clonorchis sinensis would appear to be finely granular compared to that of Opistorchis. But what will really help us identify which is which are the paired testes. If the paired testes are branched, then it's a Clonorchis worm. If the paired testes are lobulated or lobed, then it's the Opistorchis worm. These worms have vitellaria situated at the middle one-third of their bodies. Can you identify the two worms? Yes, both of them are Opistorchis adult worms. Why? Because they both have lobed or lobulated paired testes. There are two species of Opistorchis. We have the Opistorchis viverrini and Opistorchis filenius. Infections with Opistorchis viverrini are found in Thailand and other surrounding countries in Southeast Asia. That's why this parasite is also known as the Southeast Asian liver fluke. While cases of Opistorchis filenius are very high in Eastern Europe, Siberia, and the European Union. And the most common definitive hosts are cats. That's why this parasite is also known as the cat liver fluke. Opistorchis filenius. The word feline would refer to cats. So the problem now is, how will you identify if the Opistorchis adult worm you're looking at is Opistorchis filenius or Opistorchis viverrini? You have to check the testis. If the testis is deeply lobed, just like the worm on the right side of the screen, then that worm is Opistorchis viverrini. Let's try to identify the following worms. All of them have vitellaria situated only in the middle one-third of their bodies. So therefore, these worms could either be Clonorchis or Opistorchis. Check worm C. It has a dendritic or branch testis. So therefore, that is Clonorchis sinensis. So worms A and B are from the Opistorchis species because they both have lobed or lobulated testes. But it's very obvious that the testes of worm B are more deeply lobed than the testes of worm A. So therefore, worm B is Ophistorchis vivereni, while worm A is Ophistorchis filenius. There are a lot of similarities between Clonorchis and Ophistorchis from the life cycle, from the appearance of their vitellaria, and as well as their ova. Their ova are ovoid in shape, yellowish brown in color, and they have this operculum that fits on a thickened rim of the eggshell, and that thickened portion of the eggshell is called as the shoulder. The ova of Clonorchis and Ophistorchis are ovoid in shape, they are yellowish to brown in color, and they have this operculum that is fitted into the thickened portion of the shell, and that thickened portion is called as the shoulder. So to make it easy for you to remember the description of the ova of Clonorchis and Ophistorchis, I want you to compare it to a teapot. The cover of the teapot is situated on a prominent area within the body of the teapot. So that prominent area is equivalent to the shoulder of the eggshell of the ovum of Clonorchis or Ophistorchis. Do you know that there's one trematode which has the same morphology of ovum as that of Clonorchis and Ophistorchis, because it also has the thickened portion of the eggshell where the operculum is fitted onto. That's why we need to memorize another important feature of the ovum of either the Clonorchis and Ophistorchis to differentiate the ovum from that of the other trematode, which we will identify in our next lecture. So aside from the thickened rim on the eggshell where the operculum is fitted, it's also important to take note of the presence of small knob or protuberance at the posterior end of the ova or ovum. Again, the ova of Clonorchis and Ophistorchis have small knob structures or protuberance on their posterior ends. It's very important to take note that these eggs are released from the human body as embryonated ova or eggs, and they need to be ingested by the snail host for the life cycle to continue. These ova have thickened portions along their eggshell where the opercula are fitted onto, and aside from that, they also have small knob-like structures or protuberance on their posterior ends. So these ova are from Clonorchis or Ophistorchis worms. These are the first and second intermediate host of the Clonorchis and Ophistorchis worms. For the snail host of Clonorchis, we have the Paraphosarolus and Bulimus snail families. For Ophistorchis, we have the Bithynia snail family. And both these worms affect freshwater fishes, especially those from Cyprinidae group. These are pictures of fishes that are from the Cyprinidae family. Now, what are the clinical signs and symptoms of infection with Clonorchis and Ophistorchis? In light infection, most of the time, patients are asymptomatic. Should the patient become symptomatic, the patient might complain of fatigue, weakness, loss of appetite, some episodes of diarrhea, and maybe because of the loss of appetite and diarrhea, some of them will have weight loss, and there could be also abdominal pain and enlargement of the liver. In heavy infections, there is a probability now of bile duct obstruction, because you know very well that these worms prefer to stay in the lumens of the bile ducts. Thereby, they can really cause obstruction. And that will now increase the chance for the supersaturation of the bile. And supersaturation of the bile will cause formation of gallstones. And some of these worms can even enter the lumen of the gallbladder and initiate infection there, and it's referred to as cholecystitis. Aside from the bile duct obstruction, these worms can cause chronic inflammation in the bile duct, causing now fibrosis. Do you know that there are already reported cases of pancreatitis secondary to Clonorchis infection because of the invasion of the worm of the main pancreatic duct? And this one is usually diagnosed through endoscopic retrograde cholangiopancreatography, wherein the doctor might see that the tributaries or branches of the main pancreatic ducts are dilated, and it's secondary to the invasion caused by the Clonorchis adult worm. If you are diabetic and at the same time having chronic opisthorchiasis, you are at a higher risk of developing the complications of diabetes. And according to some studies, the presence of diabetes and at the same time chronic opisthorchiasis or infection with opisthorchis will increase the chance for gallstone formation. The presence of the worms in the bile duct can trigger proliferative changes and fibrosis. Now take a look at the picture of the portal triad on the left side of the screen. Letter A is hepatic artery, letter B is portal vein, and letter D, dog, is the bile duct. Now, on the right side of the screen, that's a cut section of the liver from someone who has clonorchiasis or opisthorchiasis. This is the hepatic artery, this is the portal vein, and this is the bile duct. Take a look at the worm in its center, and please do appreciate the fibrotic changes around the bile duct. The Clonorchis and Opisthorchis adult worms can also produce a carcinogen, and we call it as the nitroso compounds. I will say it again, nitroso compounds, Nitroso. So if they are present in the lumens of the bile ducts, they can actually cause the development of cholangiocarcinoma. Will there be ectopic infection in clonorchiasis and opisthorchiasis similar to fascioliasis? Remember, in fascioliasis, the metacercaria will pierce through the wall of the intestine to escape into the peritoneal cavity, to invade the liver, to gain access into the bile duct. Will that be also true to the life cycle of Clonorchis and Opisthorchis? There is no ectopic infection in clonorchiasis and opisthorchiasis. Why? As soon as the metacercaria is ingested, it will reach the bile duct not by penetrating through the wall of the intestine, but by entering through the ampulla of Vater. That's why there is no ectopic infection in Clonorchis infection and Opisthorchis infection. So let's try to review what parasites or what trematodes can cause ectopic infections, or which among them can involve the brain in the process of the infection. That's possible with Paragonimus. That's very possible with Schistosoma, and as well as the Fasciola. But there is no such thing as ectopic infection in cases of clonorchiasis and opisthorchiasis. As for the treatment, the drug of choice is praziquantel. Praziquantel can also be paired with albendazole. The pairing of the two showed good results, especially in patients that are having moderate to severe infections. Then, for the prevention, we have to treat the positive cases. We have to do health education. We have to eat sufficiently cooked fishes. We can also do salt treatment on the fish meat. And we can, aside from adding salt, we can add 3 to 6% acetic acid the 3 to 6% acetic acid will increase the diffusion of the salt into the meat of the fish, and the salt now will be able to kill the encysted metacercaria. Freezing the fish meat can also kill the metacercaria. And another means of preventing possible cases of clonorchiasis and opisthorchiasis is proper human waste disposal. And that ends my lecture on hepatic flukes. If you have questions, please don't hesitate to ask me through my social media accounts. Hello there, Fasciola hepatica with the cephalic cone.